Hey guys, in this video we're going to talk about the results from our transformation experiment and how to complete the rest of our biotechnology project. So if you're watching this video, it should be after you have completed the transformation lab and you should be checking out the results right now. Now I'm going to show you some results that students have gotten in previous years and as well as expected results, but I also want you to take a peek at some of our plates from yesterday, and Mr. Laptad should have set those out for you so you can ask him or the sub to tell you where they are and you can take a peek in class. All right, so our overarching question was, can we make bacteria glow green by inserting a gene into that bacteria to help have it produce our glowing green protein? And what our results should have been is that on our first plate with negative plasmid or our negative control, we should have seen bacterial growth just in general. We could see a lawn, we could see lots of bacteria here. It just depends on how comfortable the bacteria were and how much growth happened in the 24-hour period that we gave it. So we should see normal growth here. For the negative plasmid or negative control with our ampicillin, we should have not seen any growth here because we did not give our bacteria any ampicillin resistance gene, and the ampicillin should have killed off all of the regular bacteria. And since it doesn't have the resistance gene, it would have not been able to grow. When we have our regular Luria broth plate with, or our regular LB plate with our plasmid, we should have seen some bacteria growth. In fact, probably a lot of bacteria growth. It may be different depending on, again, how well we plated it or the conditions that it was grown in, but you should see some growth here. Now for our ampicillin plate with our plasmid, we should also see some growth with different colonies that do glow green under the black light, so you guys can check that out. I wanted to show you some of the results from previous years. So again, those were some ideal results that we saw right here. And here we see for our regular plate, it's a little bit hard to see, but there is bacteria growth on here. For our ampicillin plate with no plasmid, we did not see any growth. For our ampicillin with the plasmid, we did see our growth. There's our glowing green, and then you can see a lot of colonies on here. In fact, there's lots and lots that grew on our regular plate here. So since we're taking a look at this, I'm going to show you a couple other periods. Feel free to pause the video at any time and base your results information off of this if you would like. Here's another good view of ones from previous years. All right, so when we get to our bacterial transformation lab, you guys are going to spend today finishing up this checkpoint in the PLP. In each of the yellow boxes, you are going to justify why we did each of these steps. If you're not sure, you can use any resources like our videos or online resources. Just be sure to cite those because you will need those citations later. So justify each step. Tell us why we did each of these steps or why it's important or what's happening in our transformation lab. Once you finish that up, don't forget the lab summary at the end. Give us a shorter version of all of the steps in transformation. I don't need all of the steps detailed point by point, but just general overview. What do you think were the most important steps, and write them here. And then you're going to summarize your justifications as well. Why do we do each of these important steps? What is important about them? If you found additional sources, please cite them here. Don't worry about this section here, this section, or this section, just the orange and yellow boxes. Next up, the hypothesizing section. I really don't need you to do this since we already saw the results. You don't need to hypothesize again. This section you can basically ignore for now unless you just want to practice reasoning for your predictions, but it is totally optional and I will not be checking it on the checkpoints. All right, so here are our to-dos. Make sure you finish the pre-lab, the transformation prep work. Make sure you request feedback on the lab for the transformation lab. Once you are done with that, you make sure you've watched the Human Genome Project video, which is in our biotechnology packet. You can complete the biotech practice questions in our packet. And if you finish with all of these things and all of our other biotechnology checkpoints within this project, you can start to work on other PLT. Now, next week when we come back, we're going to be finishing up the entire biotechnology project by putting all of these procedures together into one giant lab, which is going to be exciting. So if I talk too fast or if you need to see any of this again, please go back and pause, check out the results from other classes, other years, and try to take a peek at our results as well. All right, good luck working, guys, and please email me with any questions.